So chapter 18 will talk a little bit about genomes and their evolution, and also lots about techniques that we can use to study them. So our book talks about reading leaves from the tree of life, basically looking at how different organisms are related to each other and using DNA to do so. Genomics is the study of whole sets of genes and their interactions, and because we've sequenced so many different genomes, like human genome, chimpanzee, corn, fruit fly, house mouse, orangutan, E. coli, lots of other prokaryotic organisms, we've got thousands of genomes that are completed and tens of thousands that are in progress or just considered a permanent draft because we're like, close enough. So we have all of this data, and so we need to be able to store it and analyze it. So bioinformatics is the solution to that problem. It's a whole new field that deals with storing and computational methods that help us analyze this biological data. And comparing genomes helps us understand evolutionary history and also helps us understand who's related to who in different taxonomic groupings. The Human Genome Project began in 1990 and sequencing was pretty much completed by 2003. I have a hard time convincing myself it was that long ago, but I pulled up some of the data from the Human Genome Project and the computers that were so high tech at the time. If you look at the picture now, they are definitely not the most high tech thing. So even with automation, it took a very long time to sequence all 3 billion base pairs in a haploid set of the human genome. So the Human Genome Project really gave us a drive to develop faster technology for DNA sequencing. A couple of examples of this are whole genome shotgun approach and sequencing by synthesis. So sequencing by synthesis is really just kind of what it sounds like. So the nucleotide is linked with a different dye and modified by a terminator group. And during a reaction cycle, all 4 nucleotides are going to be offered simultaneously to the polymerase that is building the strand. And after incorporation of a complementary nucleotide strand, elongation will no longer be possible because of that terminator group. And then the 4 dyes of each nucleotide can be detected using imaging. And so the dye in the terminator group are going to be cleaved, and then a new synthesis cycle will start. So the sequence of each cluster is simultaneously determined base by base by base by base by base. You think this would take a little bit of time? It absolutely would. The whole genome shotgun approach is another method. This starts with cloning and sequencing just random DNA fragments, as you can see in the picture, and then computers are used to find overlapping sequences into a single continuous sequence. So the idea with the shotgun approach is that if you aim a shotgun at it, you're gonna hit something. So that's how this gets its name. So the study of metagenomics involves discovering and modeling and understanding and looking at the molecular level, relationships between molecules that define communities and the biosphere. So if we remember back to Unit 1, a community is a group of species. So you can take DNA from a group of species in an environmental sample and sequence it and use software to sort out the partial sequences and assemble them into their specific genomes. So this is a picture, I don't remember where I found it. If I was in speech class, I'd be in trouble, but it's trying to show you that the communities that make up the microbiome in the mountains are different than what you would find in the city, and they're different than what you would find in an aquatic habitat as well. But the neat thing about this is we don't have to get just one species, we can get an entire community. So there's lots of free resources that are available to analyze genome sequences. There's a whole list of them, and I don't care that you memorize them. The one I'm most familiar with is the NIH's NCBI, so the National Center for Biotechnology Information. As the gene bank, and people are continually adding to it. It doubles its data approximately every 18 months. So it's a great resource. There's also a protein data bank that contains 3-dimensional protein structures that have been determined. And again, the cool thing about this is that they are shared by researchers and they are available free of charge. So here's just an example of what you would get if you went to the gene bank or the gen bank. So if you were there, you could search for a specific DNA sequence, you could look at the predicted protein sequence, and you could see a 3D model of that particular protein. So proteomics then is the study of full protein sets, right? A proteome is a protein set. So genomics studies genomes, proteomics studies proteomes, which are protein sets. So this can look at when and where proteins are made in a cell, or when and where proteins are made in an organism, to help us understand how these function. Systems biology then tries to look at the entire biological system. So we could look at all of the proteins that are expressed in different cancer cells and compare gene sequences in cancer versus normal cells. We talked before about microarray being used to do this. So microarray is a technique, and systems biology is like an area of study. So microarray is used to help us understand systems biology. So again, microarray, we talked about this before, but there's the gene chip, and it monitors the expression of lots of genes simultaneously, and it analyzes where genes are over or underexpressed in cells, and it can help us tailor treatments to particular patients. For example, if we know that you have a certain gene turned on or off, there may be a treatment that will help that. So here's just a few more images about DNA microarray. This is showing you you would isolate mRNA from cells of interest, add reverse transcriptase, produce complementary or cDNA, and those would hybridize to the microarray gene chip. You put it in a scanning fluorescence microscope, and you get a really cool image that shows you which genes are overexpressed or underexpressed, or which cell types express which kinds of genes. So genomes of most bacteria and archaea range from 1 to 6 million base pairs, and genomes of eukaryotic organisms are much larger. But if we think about, chromosomes and bacteria and chromosomes in eukaryotic organisms, bacteria usually have just that one single circular chromosome, and eukaryotic organisms have several linear chromosomes. But within each domain, there's not really systematic relationship between genome size and a particular phenotype. Prokaryotic organisms tend to have fewer genes than eukaryotic organisms. However, unicellular fungi don't have a whole lot of genes either. But multicellular eukaryotic organisms can have up to like 40,000 genes or more. But the number of genes is not really correlated with the genome size. So a nematode, C. elegans, has 100 million base pairs and 20,100 genes, while Drosophila fruit fly has 165 million base pairs and only 14,000 genes. So there's not a one-to-one ratio there. Invertebrate genomes can produce more than one polypeptide. What's a polypeptide again? Many peptides. Great. What's a peptide? Peptide's the bond between amino acids. So many bonds between amino acids. So that's going to be basically like a protein, right? So vertebrate genomes can produce more than one polypeptide or protein per gene because they can do alternative splicing of their RNA transcripts, where they splice the exons together differently. So really, our gene density is not very high. We've got about 1.5% of our DNA that is exons. The rest of it is regulatory sequences, introns, and then various kinds of non-coding DNA. But just because we don't know what it does yet doesn't mean it's not important. We've got some non-coding sequences that are the same in humans, rats, and mice, and other organisms. So just because we don't know what it does at this point in time doesn't mean it's not important. And since they're highly conserved or identical in so many different species, it really strongly suggests that they have some sort of function that we will discover in the future. So if we look at gene density and non-coding DNA, the DNA between functional genes includes gene fragments, pseudogenes that were probably genes before but have so many mutations now they don't really work, and then repetitive DNA, which is present in lots of different copies in the genome. This can result from errors during replication or recombination. Maybe we make a copy of a big piece that we didn't need when it got in a different spot. Repetitive DNA also includes simple sequence DNA. So we have short repetitive sequences there, like short tandem repeats if they're 2 to 5 nucleotides long. And these are kind of interesting because they can vary between individuals and can be used as genetic markers. So both prokaryotic organisms and eukaryotic organisms have stretches of DNA that can actually kind of move from one area to another within the genome, which is pretty interesting. Those are called transposable elements. Eukaryotic transposable elements can be one of two types. They can be transposons, which move by a kind of cut and paste method or a copy and paste method that leaves a copy behind and require an enzyme called transposase. Or they can be retrotransposons, which move by an RNA intermediate and always leave a copy behind. They use reverse transcriptase, so just like retroviruses, right? Retrotransposons go from RNA to DNA, so they go in kind of the quote reverse direction. So they use reverse transcriptase. So here's just a picture of a transposon being copied to a new location, or a retrotransposon, which requires an RNA piece, and then reverse transcription to make a piece of DNA, and then you have 2 DNA pieces created and inserted into the genome. So that's a retrotransposon. So many eukaryotic genes, you've only got one copy per haploid set of chromosomes. However, there are some that are found in multi-gene families where we've got copies of identical or very similar genes. These are for some things that are pretty important, like in hemoglobin. So this is good to have different kinds of hemoglobin that can be expressed at different times in development, like infant hemoglobin is different than adult hemoglobin, and also some multi-gene families have identical DNA sequences, so that if a mistake is made, it will be okay because you will have another piece, or it allows you to make a whole bunch of mRNA very quickly. So we've already talked about mutations before. Really, a mutation is just a change in a DNA sequence, and mutations contribute to evolution. The earliest forms of life likely had very few genes, just the genes that were necessary for survival and reproduction, and over evolutionary time, the size of genomes have increased. So this extra genetic material provides raw material for gene diversification. So if we have duplication of an entire chromosome set, I mean, sure, that's one way to get more DNA, right? Duplicate the entire chromosome set. So that would be polyploidy, where we've got more than a diploid chromosome number. And this happens when you have accidents of meiosis, or you could have genes in one or more of the extra sets that can diverge. They can accumulate mutations. These variations can persist if the organism can survive and reproduce. So polyploidy is pretty rare in animals, but it's pretty common in plants, especially flowering plants. We've already talked a little bit about alterations of chromosome structure, but chromosomes can fuse together. We can have unequal crossing over because those transposable elements can provide sites for crossover between non-sister chromatids. Slippage can occur during DNA replication, so part of the template region is either skipped or used twice by the replication machinery. So here's just a couple pictures to show you that. Here you've got chimpanzee chromosome 12 and 13, and the human chromosome 2 is very similar to chimpanzees 12 and 13, but fused together at a telomere-like sequence here. This is not to say that chimpanzees fuse one chromosome and you get a human. That's not what we're saying here. We'll talk more about evolution in Unit 5. And then here we're just showing you crossing over occurring unequally and creating gene duplication. So if we have exon duplication and exon shuffling, again, errors in meiosis can result in an exon being duplicated on one chromosome and deleted on another chromosome. With exon shuffling, this is when we mix and match different exons within a gene or between two different genes, and that could lead to new proteins with novel combinations of functions. So here we're just showing you ancestral genes that led to a TPA gene as it is today. So we took some an exon from epidermal growth factor, a fibronectin exon, and a plasminogen gene with a Kringle exon. And now if we look at the TPA gene as it is today, you can see it's got those pieces of exons from other genes. So transposable elements can contribute to genome evolution. Multiple copies of similar transposable elements can facilitate recombination or crossing over. Inserting transposable elements could block protein production, or they may increase or decrease protein production if they're within a regulatory sequence. And transposable elements can carry a gene or groups of genes to a new position entirely. These sorts of changes are quite honestly usually detrimental, right? They're normally a bad deal. If you have a protein in your body and you have the genes for that protein, you probably need it. But occasionally they can prove advantageous, and if that's the case, then the organism survives and reproduces. If it has an advantage over others, then we have more of that in the future. Again, we'll talk more about evolution coming up later, but this is really kind of setting the stage for us to talk about that. So we can look at different genomes to study evolution and development in organisms. If we look at closely related species, it helps us understand recent evolutionary events. And if we look at distantly related species, it helps us understand more ancient evolutionary events. It also helps us understand the great diversity of present-day life. So looking at evolutionary relationships can be done by looking at this sort of tree-shaped diagram. And again, in Unit 5, we'll talk more about how to make those and how to interpret those diagrams. But if we look at this one where it's mouse, human, chimpanzee, so mouse, human, chimpanzee, it's not saying like chimpanzee is in first place, human is in second, and mouse is in third. It's saying that mice, human, and chimpanzees share a common ancestor, and humans and chimpanzees share a common ancestor. So some areas are going to be highly conserved. They're going to remain the same or similar over time, and this helps clarify relationships among species that diverged from each other a long time ago. So bacteria, archaea, and eukaryotic organisms diverged from each other like 2 and 4 billion years ago, which is a pretty big range, but we don't know more than that at this point in time, really. But we can also look at genomic studies to confirm the relevance of research on model organisms. What I mean by that is if we are studying a medicine that we want to produce for humans, let's say to treat a heart condition, we could use a model organism like a mouse or a primate, a non-human primate, depending on what we're doing. And depending on our IRB review board's approval. But we can test it in an animal model first to see how it works and how we think it would affect human biology. So animal models are not entirely the same as humans, and I hope that someday soon we start testing things more on tissue culture, but model organisms are a decent approach to start with. So when we compare closely related species, we can assume that they'll be organized similarly, although we could be surprised sometimes. But if we look at genetic differences between two species, that can be correlated with the phenotypic differences between these two closely related species. If we compare genomes within a species, we can look at single nucleotide polymorphisms, or SNPs. Each of these represents a difference in a single nucleotide, hence the name. We can also look at inversions, deletions, duplications, and copy number variants. So this is useful for tracking human evolution and human health. And really, as a species, humans haven't existed for very long, so we have low within species genetic variation. Although if you look around at all the different people around you, there's a lot of variation within humans in a way, but there's also, genetically speaking, not that much variation. We can also use genes to look at developmental biology. So evolutionary developmental biology looks at developmental processes of different organisms, and genomic information shows us that a minor difference in a gene sequence or gene regulation or gene expression can result in a striking difference in form. So small changes in regulatory sequences of genes can lead to major changes in body form. In particular, the Hox gene is really important with this. So that was all we had for Chapter 18. Chapter 18, again, was about genomes and their evolution. So we talked about genomics, the study of genomes, bioinformatics, like how do we deal with all of this information and analyze it. The Human Genome Project got done in 2003. We described the whole genome shotgun approach to DNA sequencing, where you just synthesize random fragments and then piece them together. We also talked about sequencing by synthesis. We talked about metagenomics and proteomics and systems biology and comparisons between genome size, and we talked about how much of our DNA is non-coding DNA, but just because it's non-coding doesn't mean it's not important. Multi-gene families, where we have many copies of similar or the same gene, and how the genome has changed over time. And we looked at how we can compare genomes and single nucleotide polymorphisms, copy number variants, and short tandem repeats. So that is all of Chapter 18.